Do you have the ARS questions for me, please? Okay. The definition of BCG unresponsive or historically refractory non-muscle invasive disease includes all of the following except, you can read them, prior BCG induction and at least one course of maintenance. Remember, we're looking for all are true except. T1 high grade after induction BCG only, BCG intolerant, high grade recurrences within 6 months of the last BCG, and then positive cytology and biopsy proven CIS 6 months after last dose of BCG. So go ahead and give us your answer. Remember, all are true except. So BCG intolerant is not included. We tried to get that in there, and the FDA, the short answer is they said no. So very good. So the majority of people understand that. The next one, please. Which of the following drugs are FDA approved for treatment of BCG unresponsive CIS, BCG refractory CIS? This is if you were paying attention to Dr. Kamat's lecture. BCG, valrubicin, mitomycin, interferon, or gemcitabine. Which is the only FDA-approved drug in this space? And the hint is it was approved in 1998. Yay, everybody got it. Valrubicin, yes. This doesn't have all of my disclosures for some reason or other, but in talking about trials in the BCG unresponsive state, we have funding through the Southwest Oncology Group trial that Peter Black and Parminder Singh are heading from Roche Genentech for atezo. That's through the NCI. There's a clinical trial that we're also involved in just as a site with FKD and Viventia, and I will be just talking briefly about those. Okay, so it always helps to start with the indications for BCG, and I've listed them here, and I think Dr. Kamat outlined them quite nicely, and I too like the simple risk stratification. It's basically indicated for anybody with high-grade disease for whom intravesical therapy is appropriate. And so that's going to be high-grade TA, T1 after re-resection, multifocal and recurrent, this sort of high-risk, intermediate-risk disease that Ashish described. Standard of care for first occurrence, patients who recur after an initial induction, for whom BCG continues to be appropriate therapy. You can give another round of BCG. Oddly enough, there's really never been a properly designed randomized trial to address BCG in that specific state, meaning, say, a patient who has persistent or recurrent CIS after induction only. You can salvage a lot of those patients with additional BCG with or without interferon. And fortunately, BCG has an FDA-approved indication for CIS and high-grade papillary disease. Ashish also, I think, pointed this out, and this is really important to nail down as accurately as possible, is the status of the bladder at the 3-month mark. So that's generally 6 weeks after induction BCG. This was a secondary analysis of the BCG maintenance trial that showed quite clearly, and remember, the outcome here is survival, that patients with persistent disease at that 3-month mark have a long-term diminution in their survival probability. The flip side of that is if you have an initial CR, and we could perhaps expand that to the 6-month mark because of the sometimes delayed response to BCG, those patients clearly have a much more favorable outcome. Seems intuitive, but it's also supported by data from this randomized clinical trial. And then there was a clinical trial done by the EORTC, which looked at, it was a 2x2 study of full dose and reduced dose BCG, and then long-term maintenance at 3 years, and short-term maintenance at 1 year. And it affirmed the need for 3 years of maintenance in patients with high-risk disease. So if you're using BCG for the patient with high-grade disease, the standard of care is induction plus 3 years of maintenance, with the caveat that in the EORTC study, roughly about a third of the patients made it out to 3 years. In the SWOG study, 16% made it out to 3 years. There was no dose reduction strategy in the SWOG trial, and And obviously, a lot of those patients fell off because of recurrences. It is really important to understand what our guidelines say about what to do when the patient has had an adequate run at BCG, and you'll see that constitutes at least induction plus 1 round of maintenance, that the standard of care from our guidelines, and this is not just the AUA guidelines, is a radical cystectomy. The reason for that is because the progression risk is significant enough, and radical cystectomy has an extraordinarily high success rate in terms of long-term survival when you do it, and I'll show you a slide on this, while the patient still has non-muscle invasive bladder cancer. So when you have this conversation with the patient, you always outline the standard of care, but I think that all of us acknowledge that, number one, there's a lot of patients for whom we have a lot of trepidation about doing that because of comorbidities and risk. Patients are not lining up outside our office door to have a cystectomy. And quite frankly, there's a lot of patients for who the treated natural history, you still have a window where you may be able to cure them with a different type of therapy. That, of course, is the focus of this talk. So here's the problem, is that we do not require histologic evidence of muscle-invasive bladder cancer to justify doing a radical cystectomy, exposing the patient to that risk. And this is what the problem is that if you do a cystectomy when the patient still has muscle-invasive bladder cancer, a so-called early cystectomy, I think some of us would call that an appropriately timed cystectomy, as opposed to doing a cystectomy when there's muscle-invasive cancer, acknowledging some of the challenges with getting accurate clinical staging, is you take a big hit of about 20% decrease in their survival probability. And so we have to understand as a community what the proper timing for cystectomy and be willing, if not personally to do that, to refer them into centers where they do need that, instead of a not infrequent scenario that Tom Keene was sharing last night, I think it was, where the patient has a T1 cancer, maybe they do or don't get resected, they don't get BCG, and big surprise, you see them when they have the muscle-invasive cancer. So anyway, so that's that. Kind of remember that data in your head. So this is a phraseology used by that Diane Qualley, who's president of, just the immediate past president and co-founder of Beacon. She wants us. She doesn't want us to call this BCG failure, indicating that somehow the patient has failed. The treatment has failed the patient. So that's why it's when BCG fails the patient. What can you do? So BCG failure is induction only. Just a terminology. And those patients, as I've mentioned already, may respond to additional induction BCG, 3 months of maintenance, 3 weeks of maintenance in the setting of CIS. I like adding interferon. Remember that one of the ways that BCG works is inducing a Th1 response mediated in part by interferon gamma. So here we're giving the interferon. We know that interferon works as a single agent, but the two drugs together can be more potent in terms of stimulating the appropriate immune response, and it does allow you to reduce the dose of BCG to help manage the potential toxicity. So there are these, I've mentioned that valrubicin is the only FDA-approved drug. These other approaches, doublet therapy that Mike O'Donnell has promoted, taking a page from the medical oncologist, are all non-approved and quite frankly supported in one way or another with modest level evidence. And I think today I'll show you some data. A lot of us are rather enthusiastic about the gem-doce combination. So these would be your, aside from valrubicin, with the off-trial options that you can do now. And in my talk, there's the publications to guide you through that. And I'll show you some data. And of course, we're clinical trialists, so we try to put these patients on clinical trials whenever possible. I will skip over this because Dr. Kamat already showed you the data with valrubicin. And gemcitabine has been studied in multiple trial settings, and there's probably a publication track record of over 2,000 patients with this as a single agent. This is just one that was done by the Southwest Oncology Group in BCG, previously treated BCG patient population, and meets sort of this somewhat elusive FDA bar if this were going for a drug approval as an effective agent in this disease state of BCG unresponsive disease. As you know, gemcitabine is off patent, so you can use this as an intravesical therapy, either as a standalone drug or in combination with docetaxel. Docetaxel has been studied extensively by the group at Columbia led by Jim McKiernan. This is long-term results of their phase 1 and phase 2 trials, a modest number of patients, and a pretty healthy initial complete response. And of those patients, 18 received maintenance. This is all comers, and then this is the result with and without maintenance. And you can see that that looks like an incremental gain over valrubicin. It looks pretty good with gemcitabine, and Mike O'Donnell, as I mentioned, has looked at this in combination. This is his group's report, 45 patients, treated over 5 years, and you can see that at 2 years, roughly about a third of the patients have maintained a persistent complete response, durable complete response. And then there's a nice paper that was just published from the Hopkins group, kind of a mixed bag of patients, but also showing the potential efficacy and durability of this gem-doce combination. And we and many others are using this in the clinic, and I think so this would be kind of currently our off-trial go-to combination for patients in whom we feel like it's appropriate to not do a cystectomy straight away. A lot of exciting work done with a variety of ways to drive mitomycin C into the wall of the bladder. You can do this with electrical current, so-called electromotive. This is Savino D'Astasi's work from Italy. He's done this alternating with BCG. Synergo is a device that I think was developed in Israel. It's largely used in Europe. I think, is it used in Canada? There are clinical trials happening in the US, but this is a way of delivering heat. And then a microwave has also been used. And so there's some pretty impressive data out there using mitomycin C and one or more ways of device-assisted therapy. This is just one trial from Fred Witches published in the World Journal of Urology. So the definition of BCG unresponsive disease, Ashish gave you sort of the background of how this was evolved. This was really a request from the FDA, specifically Jonathan Jarrow, for the so-called expert community to identify a group of patients for whom BCG is no longer appropriate therapy, so-called BCG unresponsive disease. He mentioned already that it requires a minimum go at BCG of at least 6+3. We put 5+2 because we all realize that a lot of times patients don't get or tolerate the full course. Having never achieved a complete response or recurred within 6 months of the last BCG is sort of the strict definition. Patients with T1 high-grade disease after an initial induction course, I think a lot of us we're very concerned about that disease state, and cystectomy would be appropriate in that setting. I would say particularly if you've got a more larger volume or multifocal T1 high grade plus CIS, but these patients would also be eligible for these clinical trials, and the idea is that they're unlikely to respond to additional BCG. I'm not sure I have the slide in here, but there's some really nice data from Mike O'Donnell's BCG interferon sort of community study, which showed quite nicely that a third cycle of BCG infrequently was associated with a durable complete response in patients with CIS. Ashish alluded to this guidance document that now is aging, unfortunately. It was first put out for comment in November 2016, and Jonathan Jarrow would probably kill me for saying this, but I emailed him and I said, "What's up?" And this was what he told me. It's still a work in progress, and I have the email to document that this was a quote from him. I think he's being fairly generous, and oh, by the way, he was communicating with me on the day of the government shutdown, and he's a government employee, so go figure. But in all fairness, Jonathan Jarrow is a urologist. He's one of us, and he has been probably one of the handful of people within the FDA has really been our advocate. And quite frankly, I don't don't think we would be where we are today, particularly in clinical trials in this disease state, without Jonathan's really guiding hand and helping educate the decision makers in the FDA to help us. At any rate, so that guidance document sort of outlines the appropriate clinical trial design, allowing for a single-arm registration study, which we've seen this happen in advanced disease in the IO space, immuno-oncology space. We'll hear about it later today. But this is going on in this BCG unresponsive disease state. Why is that? Because we convinced them that there was no appropriate comparator. The single FDA-approved drug just really isn't good enough to be a comparator that we would be comfortable randomizing patients to that. So we have this disease state defined, we have the registration pathway designed, and guess what happens? Pharma just descends on this. In addition to pharma, our own investigator-initiated work, not mine personally, but the community is all over this space, and this is really, really exciting. So I am sure that this is not a complete list of ongoing clinical trials, and I'm going to talk about a couple of them. Gene therapy. Vicinium takes advantage of EpCAM expression on tumor cells and links that to Pseudomonas or diphtheria toxin to kill the cells. I'll talk about briefly the cold genesis study, which is another gene therapy trial delivering GCSF. I'll talk about an immuno-oncology trial. There's others going on, and then just a long list of very exciting trials going on in this disease state. And the other kind of cool thing that we're seeing is that we were a little bit concerned about, where are all these patients going to come from? And at least our experience so far, at least with some of the trials that a number of us are involved in, is that this has definitely increased the pie of patients coming in and accessible to these trials. It took us 3 years to accrue 50-some patients to the gemcitabine SWOG trial. I think the community is finishing the FKD trial in half that time and more than double the patients. And so this is really, really exciting stuff. So I mentioned this oncolytic adenovirus. This is the cell genesis. I'm not part of that trial. This was given to me by Gary Steinberg. He presented this, I think, at the AUA just most recently, as well as ASCO. And this selectively replicates in retinoblastoma pathway defective tumor cells, and it's an adenoviral-mediated gene therapy delivering GM-CSF, and you can see how it works there. And this is the summary data looking at 6-month complete response rate. And if you look at the aggregate data of CIS with or without papillary disease and initial CR of 50%, that would certainly meet that FDA bar if it shows to be durable over time. This is the ad interferon gene therapy trial, so also a replication deficient adenovirus delivering interferon. This is over a decade of work by Colin Denny and his colleagues at the MD Anderson Cancer Center, and FKD is a Finnish company that acquired the product and has been very supportive and working exclusively with the SUO-CTC to do a phase 2 trial, and the phase 3 trial has this same standard single-arm registration pathway design. And the key thing is that we have to prove in these clinical trials efficacy in CIS. So if you think about it from a single-arm standpoint, what the FDA has been adamant about is you have to show treatment efficacy of a cancer. And then the only one, and otherwise if we're just treating resected Ta-T1, it's adjuvant. And there's really no way to know exactly what your treatment effect without a comparator. So you have to show efficacy in CIS. This is the trial design. I'm kind of running low on time, so let me just kind of cruise through this. This is the SWOG trial, 1605, led by Peter Black and Parminder Singh. It's looking at atezolizumab, one of the IO agents in this same exact disease space, and it builds on the efficacy in advanced disease. It utilizes or takes advantage of the fact that PD-L1 is expressed in non-muscle invasive bladder cancer, and there's the hypothesis for you. This is the trial design. Patients get treated every 3 weeks, the standard cystoscopy evaluation at 3-month intervals, and we're powered to look at the complete response rate 6 months post-TURBT. This trial is open and accruing, but still early in the trial history. So just to finish up, the BCG unresponsive state defines a patient population for whom there's increased risk of progression, for whom radical cystectomy is the standard of care, but for whom BCG is no longer appropriate therapy. So I've given you sort of a list of off-trial options with chemotherapy, intravesical therapy, and then this exciting clinical trial disease state. And if you have time, I can answer questions, but I do know that there's a break coming up. So thank you very much.